Hello everyone, welcome back to my channel. In today's video, I am starting a long-term microbial growth experiment, and this is day 1 of the series. Over the next several days, I will be adding one new substance each day into this jar, and we will observe how microbial life slowly appears and changes over time, both with the naked eye and under the microscope. So this video will act as a baseline, the clean starting point. Right now, I am collecting normal tap water. Tap water can already contain microorganisms, spores, or other organic particles. So before starting this experiment, I want to reset everything as much as possible. This is important because if we don't control day 1, we won't know what changes are caused by the things we add later. Now I am boiling the tap water on the gas stove. Boiling water is one of the simplest sterilization methods available at home. At high temperatures, most bacteria, protozoa, algae, and even many spores are destroyed. I allowed the water to cool down naturally and transferred it to this clean jar. I am not adding anything to it yet. No soil, no sugar, and no plant matter. This is just boiled water poured in a clean glass jar. This jar will stay sealed and untouched from now. From tomorrow onward, I will start introducing different materials one per day, and we will see how microbial growth reacts. Each day in this series, I will add one specific substance. Wait 24 hours, then observe the changes under the microscope. Some additions may cause rapid microbial growth, some may do almost nothing, and some may completely change the ecosystem. This is basically a mini microbial universe developing day by day. I am placing a small drop of this water directly onto a clean glass slide. Since this is already water, I am not adding any extra liquid. The goal is to observe the sample exactly as it exists inside the jar without any modification. Now I am gently placing a cover slip on top of the sample. This helps spread the water evenly and also prevents evaporation while observing under the microscope. I am being careful to avoid air bubbles because bubbles can sometimes look like microorganisms at low magnification. Let's begin the observation at 50x magnification. At this level, we are mainly looking for large particles, debris, any obvious movement at the moment. This field looks mostly clear. I don't see any visible life forms or large structures. This is expected because the water was boiled before being stored. Now I am observing the water sample at 100x magnification. At this level, I can see a few tiny structures in the field of view. They appear as small dots or short shapes, but I do not see any movement. There is no swimming, no directional motion or vibration or twitching. These structures are most likely dead bacteria or bacterial fragments. Even after boiling, the physical shapes of bacteria can remain, although the cells themselves no longer alive. So what we are seeing here is cell debris, not living microorganisms. This is an important observation because shape alone does not mean life. Movement and activity are key indicators. Now I am increasing the magnification to 450x. At this magnification, bacteria are easier to resolve. I can see very small rod-like and dot-like structures. But again, there is no active motion. They are completely static. Living bacteria usually show either Brownian-like movement, slow drifting, or active swimming. None of that is visible here. This strongly suggests that these are dead bacteria or non-living bacterial remnants killed during the boiling process. Boiling destroys the internal structures required for life, but the outer cell shapes can still remain visible under higher magnification. So this concludes day 1 of microbial growth jar experiment. From day 2 onwards, things will start getting interesting. If you want to see how microorganisms slowly appear, multiply and interact, make sure to subscribe and follow this playlist. Tomorrow I will be adding the first substance into this jar. Let's see how life begins.